Oil flowers and oil-collecting bees, Malpighiaceae and their pollinators. In large areas of South America, as here in the state of Sao Paulo, only remnants of the original primary forest are left. However, these areas are also still very rich in species today. The crowns of tall trees project above several lower stories of woody plants. In this richly structured vegetation, woody lianas use freestanding trees as supports to reach the vital sunlight. Members of the Malpighiaceae are among them. In this family, approximately 950 species are known alone in the neotropics. In the Brazilian Mata de Santa Genebra Nature Reserve near Campinas, most species of the Malpighiaceae start flowering in February, including Stigmaphyllon lalandianum, whose densely foliaged festoons are now covered with a profusion of light yellow flowers. This species belongs to the group of the oil flowers, a type of blossoms that have developed a most unusual and fascinating relationship with their pollinators. The flowers are completely devoid of nectar, but yet they are pollinated by bees that are attracted by a special fatty oil. The bees selectively search for this oil. Relatively few genera of bees share this peculiarity. They belong in part to the solitary Centridini, a tribe of the family Apidae. These oil-collecting bees vary considerably in their body size. The bumblebee-sized, somewhat ponderous species, as here Centris colaris, prefer to fly along the periphery of the liana curtain in their movement from flower to flower. However, the smaller species are much more versatile flyers and manage to reach even the flowers deeply hidden among the foliage by hectic flight maneuvers. At least 8 different species exploit Stigmaphyllon's oil. This tireless collecting activity has a single purpose: to provide food for the bees' offspring. It does not serve as food for the adults themselves. Only the females care for the brood. Therefore, the females alone engage in gathering food for their larvae. The males merely play a role as sexual partners. During oil collecting, the animal's ventral thorax becomes dusted with pollen. This pollen adheres to the stigmas of other flowers during subsequent visits. A flower reaches approximately 15 millimeters in size. There are 5 long-stalked petals. The middle, superior petal, termed the vexillum, provides the bee with a foothold. The back of the flower bears the oval oil glands or elaiophores, which are arranged in pairs as special appendages of the calyx segments. 4 twin glands alternate with the petals, but the lowermost, median pair is absent. In the course of evolution, the elaiophores have most probably originated from extrafloral nectaries, which are still present on the green leaves of the plant. These glands are epithelial elaiophores. That means that the oil is secreted by a palisade-like epidermis. This secretion lies concealed beneath a cuticular blister. Only after pressure has been exerted by a stroking bee leg does the cuticle burst at a preformed weak spot and an oil drop oozes out. After the flower bud has opened, the calyx segments with the elaiophores bend forward directly between the claws of the petals and thus become easily accessible to the bees. The densely arranged reproductive organs of the flower are difficult to differentiate visually. 2 whorls of 5 stamens each make up the androecium. The gynoecium has 3 stylodia and stigmas. Even on the buds, the elaiophores are clearly visible. They are already filled with oil the day before blossoming. Anthesis of a single blossom lasts 2 to 3 days. A bee, here for example Centris scolaris, is seen harvesting the oil with her fore and middle legs. The yield is transferred to the plumose trousers of the posterior legs and is finally unloaded into the brood cells as a larval food. These storage and transporting organs are called scopae. Fourfold slow motion allows observation of how a bee approaches a flower with her antennae stretched forward. Due to the bee's weight, the flower drops a considerable distance. The oil is removed from each twin gland by 3 scraping strokes of the legs on average. The bee alights and tightly grasps the claw of the vexillum with her mandibles. The collected oil initially remains in the densely pilose areas of the fore and middle legs. This scanning micrograph shows the foreleg of a Centris species. The oil collectors are located on the metatarsus of the front and middle legs. From there, the collected oil seeps into the plumate fringe on the outer flank, visible above, where it is temporarily stored. A comb of elastic spatulate chitinic setae comprises an oil scraper. 3 large broad setae are located opposite the comb. As seen here, these are absent from the middle legs. This bee performs a purely oil-collecting flight, as is indicated by her moist, darkly shiny scopae. The liquid is passed from the forelegs to the middle legs by squeezing it out between the bent legs and is finally pasted onto the inner flanks of the posterior legs. Here again, the oil is squeezed out and then backloaded onto the scopae of the hind legs. Epicharis rustica, a somewhat smaller species of the same affinity, also displays a corresponding collecting behavior. Her scopae are yellowish in color and carry a mixture of oil and pollen. Her leg pairs scrape alternately, to the right and then to the left, etc., across the elaiophores. Although flower buds are also intermittently visited, enough pollen adheres to the bee. To demonstrate this, a female was immobilized. Her ventral thorax is heavily powdered with pollen. The scopae conspicuously contrast to this due to their fox-red coloration. Their special coloring probably plays a role as a cue in their mating ritual. Note these animals' long tongues. In this slow-motion sequence, all steps of oil collecting and backloading can be observed once again. Positioning, grasping the vexillum and landing, oil scraping, backloading. The bee spreads her scopae backward to secure the load. To recover from this energy-consuming collecting flights, the bees exploit sources of nectar to nourish themselves, such as this Arabidea species of the Bignoniaceae. She spreads the oil from her forelegs to the inner flanks of her scopae. In this action, Epicharis does not differ from the other Centridini. Once again, the backloading procedure. It can be regarded as modified grooming behavior. To keep her legs available for pollen transfer to the scopae, the bee hangs onto a leaf by clinging to it temporarily with her mandibles. Here an Epicharis female is grooming her body. Despite her small body size of only 15 millimeters, Centris similis is also a legitimate stigmaphyllon pollinator. Compared to the larger species, however, she is capable of more versatile flight maneuvers. While orienting herself above a flower, here in slow motion, the bee can obviously check the latter's oil content. Perhaps her antennae determine whether it will be worthwhile to exploit the flower's oil based on the latter's odor. Here the backloading procedure can be observed 3 times in sequence. Centris varia, the tiniest species observed. She performs especially hectic flight maneuvers. She also transfers the yield to her scopae with similar skimming movements, and occasionally spreads her hind legs upward. Centris varia prefers to visit the buds of the flowers, perhaps because the open flowers have already been emptied as a result of the strong competition by larger species. Because the vixillum is not yet available for clinging, she must use her posterior legs for support on landing. Since the petals do not yet obstruct our view, the scraping of the elaiophores can easily be observed. These oil-collecting bees differ considerably in their choice of breeding sites. Some Centris species make use of tunnels burrowed by coleopteran larvae in old wooden posts, but most species dig their own burrows for rearing their larvae. About 110 miles west of Campinas, there is an experimental station of the University of Ribeirão Preto. Here, the breeding behavior of wood-nesting Centris species is investigated in detail. To this end, short pieces of hollow bamboo cane are offered to the bees as a nesting aid and are readily accepted by them. Here a Centris vittata female has chosen her breeding place. She constructs a series of brood cells in the tube by applying a mortar-like mass of oil and sand grains. An occupied piece of bamboo has been split lengthwise. This allows us to look at the nest. A layer of dried oil covers the outer wall that consists of stabilized sand. The wall's upper half has been removed cautiously to be able to look into the brood cells. In the youngest brood cell, a larva is floating in a nutrient paste of oil and pollen. The larvae of the following, older cells are, with one exception, more mature. The original primary cell is located at the posterior end of the tube. Its larva will be the first to pupate. The cell's heterochronous establishment makes pupal stages of differing lengths necessary. Only in this way can the youngest individual in the foremost cell hatch first and to leave the nest through the entrance. There are residual primary forest areas near the experimental station. The Malpae Banisteriopsis muricata grows in this area. Its luxuriant inflorescences glow in rose and white colors. A horizontal axis bears flowers of different ages. During anthesis, their colors change from crimson to white. The reproductive parts of the Banisteriopsis flower resemble those of stigmaphyllon. However, in this case, they are divided into 2 functionally different complexes. The one directed downwards provides fodder pollen for the bee and its young. This complex consists of 1 style and 8 stamens, 5 of which bear large glands. These glands produce an oil that possibly enables the pollen to better adhere to the bee. The second complex is the reproductive one. It is formed by one pair of upright, long-stalked stamens positioned on either side of the vexillum and each accompanied by a style with a small stigma. The Banisteriopsis flower also exhibits 4 narrow twin oil glands when viewed from the back. They are absent on the 5th median calyx segment. The notch on the sturdy vexillum claw has dark necrotic scars. They reveal that it has already been grasped by bees. These sequences, taken at normal speed, illustrate how zealously and thoroughly the bee works in inflorescence. Although Banisteriopsis is visited by other species of Centridini, the details of the collecting procedure will not be discussed further. The oil harvesting procedure is the same in all these species. It has only recently been discovered that flowers offering fatty oil to pollinators, although they represent a minority compared to the classical rewards of nectar and pollen, nevertheless play an important role in certain plant communities, both in terms of ecology and in number. Apart from the extraordinary collecting behavior shared by these bees, the coincidence of distribution areas of both partners reflects their coevolution. Besides the Malpighiaceae, pollinator attraction by floral oil has also evolved in other, taxonomically very distant families. This allows the conclusion that we are dealing with a strategy that has independently evolved several times. Oil flowers and oil-collecting bees, pollination of Angelonia hirta. In many regions of the tropics, the indigenous forests have been clear-cut and have been replaced by plantations and pastures, as is the case on this northeastern Brazilian hillside. Nonetheless, a plant community rich in species has survived. This species richness corresponds to a high diversity of pollinators. Among the resulting relationships, special symbioses have evolved between certain bees and their host plants. One of these symbiotic interdependencies is particularly fascinating. In this partnership, the flowers do not offer nectar to their visitors, as is the usual case, but instead a fatty oil. Consequently, these flowers are called oil flowers. Angelonia hirta of the snapdragon family, Scrofulariaceae, is a perennial plant that grows on moist, open sites and along thicket edges. Angelonia blooms in the rainy season between February and August. The flowers are borne on terminal racemes of the main shoot and its branches. Here a small bee of the genus Paratetrapedia is gathering oil. This bee is too small to pollinate the flowers. Instead, it is an oil thief. The flowers open successively from the bottom towards the top. Usually 4 to 5 blossoms are open at the same time. The zygomorphic corolla is pendantly borne on a delicate pedicel. As a consequence, the gaping, ornamented throat opens laterally. The median corolla lobe differs from the other lobes in its shape and the presence of a white spot. The hidden entrances to the 2 oil vessels are at the lower end of the throat's cavity. The reproductive organs lie under the ridge of the throat. During the flowering period, which lasts approximately 6 days, The anthers split open before the stigma becomes receptive. The two anterior anthers are the first to release their sticky pollen. On the second day, the two posterior anthers also open. Thus, pollen is available for an extended period. In this strictly male stage, the style and stigma are still nearly invisible from the outside. Only on the fourth or fifth day does the style become receptive. The anthers are now nearly empty, shrunk and withdrawn, whereas the style has grown up to the previous level of the anthers. As a consequence of these rearrangements, the stigma can only be pollinated by pollen from other flowers. Here a flower is seen from behind. The dark oil-containing pockets contrast with the bottom of the throat. The bees gather the oily secretion with their legs. They do not gather the oil for their own nourishment, but exclusively as food for their offspring. In addition, the oil serves to cement and waterproof the walls of their earthen brood cells. A tangential longitudinal section through the flower allows us to look at one of the pouches. The oil-secreting area, termed the elaiophore, is the dull green cushion visible on the inner anterior wall of the pouch. The entrance of the pouch is only 1 millimeter wide, just enough to accommodate a bee's leg. The oil does not fill the entire pouch, but instead saturates the cover of glandular trichomes that produce it. They are green because they contain chlorophyll, and they continuously secrete the oil during anthesis. Here another view of the intact rear wall of a flower. If the rear wall is cut away, the front wall of the two pouches with their elaiophores becomes visible. The twin pouches only allow one pair of legs, the forelegs, to be used for collecting. Other oil flowers, such as those of the Malpighiaceae, have 4 oil glands and allow bees to collect the oil with 2 pairs of legs. A single elaiophore contains about 7,500 glandular hairs. Their secretion accumulates in the capillary spaces in the hair cushion. The slightly foamy oil is squeezed out of the glandular cushion by upward stroking movements. Approximately 0.6 milligram of oil are produced per flower. The bulging emergence of the lower lip and the narrow entrances to the pouches force the bees to take on a collecting attitude that optimizes the pollination of the flower. At least 5 bee species exploit Angelonia hirta and pollinate it in the process. They are solitary, and all of them belong to the Centridini, a tribe of the family Apidae. Their body size varies considerably. Here, for instance, the bumblebee-sized, somewhat clumsy Centris obsoleta of the subgenus Melacentris. Only the females collect oil because they alone take care of the brood. The orange patch on the bee's back is due to the pollen of a nectariferous passion flower she visited some time before. In flight, the collected oil is passed to the densely plumose shanks of the hind legs, the scopae, which serve as a transporting organ. In this slow-motion sequence, the oil harvesting procedure is visible in more detail. The bee alights on the flower while embracing its lower lip with her middle legs. After the wings have come to rest, the forelegs are inserted simultaneously into the 2 pouches. They now start scraping off the oil. In flight, the bee then transfers the oil to the scopae of her hind legs with her middle legs. While collecting, the bee is forced to press her head against the anthers. In the process, the pollen adheres to the bee's forehead. The flower sinks downward as a consequence of the bee's weight. The bee slips her forelegs alternately into the pouches 2 or 3 times. By means of an upward pressing movement, she squeezes the oil out of the glandular cushion. One sees the oil shining on the scopal surface. smaller bee of the subgenus Centris with a rust-colored thorax. She clings to the lateral lobes of the corolla with her middle legs. This bee species has not yet been taxonomically described. Here an individual of Centris fuscata of the subgenus Trachina with chestnut-brown abdomen and eyes. The middle legs also perform collecting movements outside the pouches, but they do so in vain, as only 2 elaiophores are present. Centris enea of the subgenus Centris also belongs to the legitimate visitors of Angelonia. This species can be recognized by its black eyes and a black transverse band across its abdomen, which has a green metallic luster. This individual's forehead is also dusted with a whitish pollen. Before alighting on a flower, she moves into the proper landing position. She hovers briefly. She appears to check how much oil is in the blossoms with her antennae, possibly by sense of smell. The same species shown in slow motion. The forelegs move alternately. Centris enea also performs futile collecting movements with her middle legs. While performing oil-scraping movements, the knees of the forelegs almost reach the head when in their highest position. Between 10 and 100 flowers must be visited to fill the scopae completely. Then the bee returns to her nest, where the oil load will be stripped off the scopae into the brood cells. The foraging flights last from 7 o'clock in the morning until late afternoon. Intermittently, oil and pollen foraging sessions are briefly interrupted by feeding flights, during which the bees take nectar to renew their own energy supply. The bee's active movement among the racemes of different plants ensures cross-pollination. While hovering, the bee often swings her body around its longitudinal axis. The head compensates for these movements. We now observe the backloading procedure, which is performed during the flight, in greater detail, again in slow motion. First, the 2 extended forelegs are simultaneously wiped over the outer flank of the middle legs. Next, the middle legs are individually and successively passed between the 2 hind legs, which are pressed together. The process is repeated once more. From fore to middle leg, from middle leg to hind leg. Backloading during flight can be interpreted as modified grooming behavior. The oil harvesting behavior is closely connected with structural characteristics of bees and their flowers. Fatty oil offered as a floral reward occurs in members of several taxonomically very distant plant families. As is evident from this circumstance, oil-based bee-flower relationships have evolved independently several times. In the course of evolution, this tight link resulted in these oils being just as indispensable for the bees as the bees' pollination activities for the plants. From fore to middle leg, from middle leg to hind leg. Backloading during flight can be interpreted as modified grooming behavior. The oil harvesting behavior is closely connected with structural characteristics of bees and their flowers. Fatty oil offered as a floral reward occurs in members of several taxonomically very distant plant families. As is evident from this circumstance, oil-based bee-flower relationships have evolved independently several times. In the course of evolution, this tight link resulted in these oils being just as indispensable for the bees as the bees' pollination activities for the plants. Oil flowers and oil-collecting bees, yellow loosestrife and hair-legged mining bees, Lysimachia and Macropis. The banks of the backwaters of the Middle Rhine River are shaped by specific ecological conditions. Due to fluctuations in the inflow rates, the water level varies considerably in the course of the year. Limited by the respective maximum height of the water, more or less extended flood plains with specific riverine plant communities have formed. Here a layer of herbaceous plants adapted to changing water levels grows alongside high water tolerant trees. Reeds occupy large areas of the riverbanks, but also conspicuous perennial herbs such as the purple loosestrife grow here. On the alluvial land among the reeds, a peculiar and fascinating symbiosis can be observed. The interrelationship between a flowering plant and a wild bee species exclusively adapted to this plant. The yellow loosestrife, which belongs to the primrose family, produces a fatty oil in its flowers that is used by the bee to feed its young. Since, in this case, fatty oil is offered as a reward, the blossoms of the loosestrife, Lysimachia vulgaris, have been assigned to the group of the so-called oil flowers. They lack true nectar. Lysimachia frequently forms thick stands by means of stolons. These are visited by their pollinating agent, the hairy-legged mining bee Macropis labiata. This reciprocal relationship was discovered not long ago by Professor Vogel from the University of Mainz, Germany. He documented the morphological and functional aspects of these two partners. First, the structural traits of the Lysimachia flower shall be demonstrated in detail. The inflorescence represents a richly branched, pyramid-shaped open panicle. The profusion of flowers is additionally amplified by so-called corroborating shoots that arise from the base of the main inflorescence. Open blossoms are distributed over space and time. This results in a continuous blooming period that lasts for several weeks. From end of June until August, the females of Macropis labiata are busy collecting oil and pollen on sunny days. They do this exclusively on Lysimachia. The collected oil-pollen mixture is very rich in calories. However, it does not serve as food for the adults, but is only destined to nourish the young that are reared in the subterranean brood cells. The female collects the oil with her legs. To this purpose she dabs and brushes over an oil-secreting zone in the center of the flower. In the process, the animal's underside is copiously dusted with pollen. Thus, pollen collection is rather a side effect of the active oil-collecting behavior. Occasionally, hovering flies also approach the flowers. However, they are only interested in the pollen, which is dabbed from the anthers with their proboscises and eaten. In addition to Lysimachia vulgaris, about 200 other species of this genus exist worldwide. Their center of distribution lies in Eastern Asia. About 2/3 of the species offer only pollen as a reward for their visitors or are nectariferous. A representative of the nectar-bearing flowers, for example, is this Mediterranean species, Lysimachia ephemerum. This plant has unspecialized bee flowers and is therefore visited by numerous diverse species of wild bees. Bumblebee workers, too, take the nectar with their proboscises. Only 1/3 of all Lysimachia species have oil flowers. According to the strict dependency of the bee genus Macropis, their distribution area, extending over Eurasia and North America, coincides with the occurrence of Macropis, of which there are 15 known species. Returning to our oil flower, we shall now consider its structure in more detail. In this frontal view, the regular, symmetrical position of the 5 petals is evident. The petals are connated at the very base. The 5 stamens are fused and form a central cone, the filament tube. In accordance with the flower's upright ascending orientation, the stamens are unequal in length. The 3 lower ones are longer than the 2 upper ones. The mobile anthers, pointed inwards, release the sticky yellow pollen. The style, with a punctiform moist stigma, extends above the level of the anthers. The filament tube is basally beset with a dense cover of 2,000 to 3,000 glandular hairs. This zone is called the elaiophore. It secretes a fatty oil that mainly consists of esterified hydroxy fatty acids. The cover of finely stalked glandular hairs extends up to the free parts of the filaments. Oil secretion begins in the bud. A Macropis female has alighted on a flower and starts collecting oil. The so-called scopae, long-haired storage and transport organs, are located on the hind legs. In the present case, they are slender because the bee's collecting flight has just begun. The tarsal limbs of the fore and middle legs are furnished with rubbing surfaces, which are composed of antler-shaped absorptive setae. These initially take up the oil by capillary action. Here, the collecting procedure is shown in slow motion. The bee's attention is exclusively directed toward the oil-producing elaiophore. The fore and middle legs dab and wipe the entire glandular surface, and in the process they brush the oil, which is bound by capillary forces, out of the hair cover. Since the bee must simultaneously cling to the flower, the individual pairs of legs do not move synchronously, but work alternately. The long and densely plumose hind leg pair with the scopae remains inactive in this process. When the scopae are nearly saturated with the oil-pollen mixture, the hind legs are stretched upward strikingly. To gather oil effectively, the bee turns around the filament tube. In doing so, her hirsute ventral side is simultaneously dusted with the pollen. During certain phases of nest building, the animals collect only oil. They then use it to impregnate the earthen walls of their brood cells. This female is carrying an oil-pollen mixture as food for her larvae. The oil moistens the pollen load, lending it a dark color and a glossy surface. At the end of a collecting flight, a bee's full burden weighs about 10 milligrams. 7 of such flights on average are necessary to produce a loaf for a single larva. This amount constitutes about a day's work of a single bee. Here, the intense brushing movements of the fore and middle legs can be seen. The backloading of the yield onto the scopae of the hind legs mainly takes place while on the wing between 2 flower visits. But occasionally, backloading occurs while the bee is still on the flower. To this end, oil and pollen are smeared from the forelegs onto the flanks of the middle legs. The middle legs then paste it on the scopae. This transfer occurs separately on each side of the body. It can be regarded as modified grooming behavior. The females construct their nest in the soil, usually not far from the oil host plants. The entrance is hidden. A small heap of excavated soil marks it. From the entrance, a ramified tunnel system leads downward, and each of its branches ends in one brood cell. These bees are solitary, but they often nest gregariously. Fertilized Macropis females usually spend the night near the brood cells in the nest, but virgin females and the males normally remain in the blossoms of Lysimachia overnight. Still a bit stiff from the low nocturnal temperatures, a female is awakening in the sunshine. Prior to start collecting flight, she grooms herself with her limbs. Now the scopae's own coloration becomes obvious on the hind legs. The silvery white of the tibia contrasts well with the black of the basitarsus. This pattern probably exerts a signal effect during mating. Before the female starts her first collecting flight, she takes pollen from the anthers and eats it. For their own energy supply, females and males take nectar with their short proboscises. Their nectar hosts are easily accessible flowers of various species. Here, a female of Macropis fulvipes, the second Central European species, is seen visiting Geranium sylvaticum, the wood crane's bill. These recordings, taken at normal speed, show how hectic the movements of the bee are, but do not allow us to perceive any details. Prior to alighting, here again in slow motion, the hovering animal directs its antennae towards the flower and balances itself to achieve the right landing position. Oil and pollen are backloaded independently and in different manners. The pollen is taken up by special combs. To this end, the forelegs are crossed and brush each other. The pollen thus removed, together with pollen from the bee's ventral side, is taken up by the middle legs and then packed onto the load already present in the scopae. Thus, the scopae attain a rounded, smooth surface. Since the pollen is never brushed off completely, enough of it always remains on the thorax to pollinate the flowers. Only the females gather the oil and care for the young. The male's only function is a reproductive one. Copulation usually takes place around the oil host. Here a male appears briefly. He has distinctly longer antennae. Here a male is attempting to copulate. However, the female repulses the male. During hovering, only the oil is selectively backloaded. The oil is not transferred to the inner flank of the middle legs, as is the case with pollen, but is transferred to their outer flanks. From these, it is then smeared onto the inner and outer flank of the scopal oil pollen mass. Flowers that have freshly opened before dawn are particularly rich in pollen and oil. These are therefore eagerly desired, and the bees often force themselves into the narrowly gaping corolla. The creamy scopal load is then in danger of being stripped off by the pointed corolla lobes. This is probably the reason why hind legs with fully loaded scopae are stretched upward in a grotesque manner. The obvious food ball on the posterior legs has given the bees their German vernacular name, Schenkelbiene, thigh bee. Compared to the common floral rewards, nectar and pollen, the production of oil is relatively rare. It occurs independently in members of a number of non-related families. Most of what we know about this oil-based plant-bee relationship was learned in the southern hemisphere. Oil flowers have been described from a number of plant families there. The interdependence of Lysimachia and Macropis is the only one of its kind known from Europe. It proves that such a pollination strategy may also occur in our latitudes. The obligate mutual dependence of these partners in pollination results in the circumstance that their joint survival will only be guaranteed where the requisite ecological conditions are favorable to both the bee and its host plant. If only one of these two partners is missing, the survival of the other is no longer certain.